Today, we'll be talking about PCR techniques in biotechnology. So what is PCR? PCR, or Polymerase-Chain reaction, is a technique used to replicate DNA and amplify sequences of DNA in vitro. PCR works in 3 steps, called denaturing, annealing, and elongation. The first step of PCR is called denaturing. The denaturing stage occurs at a very high temperature, around 94 to 95 degrees Celsius, and breaks the hydrogen bonds between the double-stranded DNA. This allows replication of the DNA without helicase. The annealing stage is where the primers bind to the beginning of the DNA sequence that is being amplified or replicated and occurs at 48 degrees Celsius to 72 degrees Celsius. The primer is required for DNA polymerase to bind and extend the strand. The final stage is elongation, where DNA polymerase extends the DNA primer to make a new strand of DNA. It occurs at the temperature at which the DNA polymerase functions best. Each cycle of PCR uses the newly created DNA as a template strand, which is where the chain reaction part of PCR comes from. Each cycle results in a rough doubling of the DNA. One error that can occur during PCR is primer dimers. Primer dimers occur when the primers bind to each other instead of the template DNA, which results in the primers being amplified and not the template DNA. Polymerase will extend the primer dimers, which makes a combined version of the primers. This can reduce the amount of amplification from the template strand of DNA. The materials for PCR are a thermostable DNA polymerase, such as Taq polymerase. Primers for the DNA sequence of interest lose nucleotides in certain ions and buffer for the polymerase. For DNA strands with many cytosine and guanine, DMSO or glycerol should be added to the reaction, as there is one additional hydrogen bond between cytosine and guanine. So how can PCR be used? One of the ways PCR can be used is for in vitro replication of DNA to amplify the sequence. Another way that PCR is used in biotech is to amplify a gene of interest. The gene of interest, after being purified, can be put into a plasmid, which will allow you to create a product from that gene in bacteria. PCR can also be used to amplify a gene from a genome of an organism.